without transition step. Then you add these together, 6 ATP right here, 8 right here, 24 right here. You add up together, 38 ATPs. Okay, now somebody gonna say, Dr. Sheng, what you're saying is wrong. What I learned in my biochemistry class is 36 ATPs. There's some people come to me after class and say you're wrong. What's the difference? This is bacteria. When you learn your biochemistry, is human being, animal, or plants, that is eukaryotes. What's the difference? What are these happen in cytoplasma membrane for bacteria? What is going to happen in a eukaryotes? Mitochondria, is that right? So, when you go into the mitochondria, you go to the park. Okay, anyway, you go national park, maybe it's free. When you go to see a museum or cinema, do you need a ticket? You need to buy a ticket, is that right? That ticket worth 2 ATPs. Okay, therefore, there is missing 2 ATPs because this is nature passing mitochondrial barrier. Okay, like you buy a ticket to go through a gate. That's why in yeast and molds, in eukaryotes, it's 36 ATPs. In bacteria, so here we talk about the yeast, molds, because eukaryotes, and this is what we talk about is bacteria, and of course it's archaea. We don't mention archaea here. So that's the difference. Okay, you need to know. If in the exam I'm going to ask you, how many ATP going to generate going through complete aerobic respiration for a yeast and the molds, for chlamydia, let's say, or Candida albicans? Just give an example. You tell me that 36 ATPs. Why? Because they need passing the mitochondrial barrier, like you buy tickets. Okay, go to a concert. So this area is what you should know. Everybody should know the calculation. Some of you are sophomore or freshman or haven't been learned biochemistry. You don't have to worry about all these chemicals. The major one you should know: glucose, pyruvate, acetyl-CoA you should know, and you should know the location where they're going to happen. Is NAD reduced to NADH2, FAD reduced to FADH2. That is talk about electron transport chain, which is chemosmotic, and they are generating more ATP compared to the substrate level phosphorylation. Okay, so these things you need to know and how to calculate. Our slides has a one concluded table at the end. It's very good, and you should read it. This gives you conclusion of each stage. And these type of the question could make it a little bit complicated. You tell me how many generated through substrate level phosphorylation. If I have this question, they make you a little bit tricky. That means 2 generate from here, and 2 comes from here. So that's 4 ATPs going through substrate level phosphorylation. Then how many going through oxidative phosphorylation? 38 minus by 4 equals 34. Okay, you look at the slides, they are concluded. We'll go over it next week, Tuesday. Okay, I want you guys to study at least here. If you need extra points, you do everything except the enzyme. I don't care about the enzyme. It's not a biochemistry class. I want one of you guys will be volunteer come here to show everybody how to calculate 38 ATPs. Can you prepare this in the weekend? You can have your notes. If you like what I did, that's okay. So next Tuesday, the first thing is somebody come here. Draw on the blackboard how you calculate the 38 ATPs, which means give you a chance to practice by yourself. Because when you wrote like this, compared to you wrote like this, a little bit difference. If you can wrote like this, there's no problem for you to do in the exam questions. Okay? And then we're going to go over the slides, because we have something we didn't mention. I'm going to look the slides and what the key is at. And then we move on to the DNA replication. Okay? So that's going to be the procedure. Okay, so that's all I have for the class.